Bismillahirrahmanirrahim. Assalamu alaikum. My name is Awais Sadat and today's topic for my presentation is stages of truth development. Actually, truth development is the continuous process, but for the sake of perception, we divide it into three processes: bud stage, cap stage, and bell shape. These are named according to their appearance. For example, if it looks like a bud shape, then it is the bud stage. If it looks like a bell shape, then it's the bell shape, and so on. Introduction. Tooth formation occur in the 6th week of interuterine life with the formation of primary epithelial band. At about 7 weeks of primary epithelial band divides into a lingual process called dental lamina and a buccal process called vestibular lamina. Vestibular lamina form the vestibule while the dental lamina form the actual teeth. The deciduous teeth arises from dental lamina. Later, the permanent successor arises from its lingual extensions and the permanent molar from its dental extensions. Now, stages of dental development. As I told you that it is divided into three stages: bud stage, cap stage, and bell stage. The names are given, as I told you earlier, that the names are given according to a base of appearance. For example, if it looked like a bud, we name it bud stage. If it looked like a cap, we name it a cap stage, and same is the case with bell stage. Now, bud stage. It is the very initial stage of tooth formation. I repeat, very initial stage where the enamel organ resembles the small bud. During the bud stage, the enamel organ consists of peripherally located low columnar cell and a centrally located polygonal cell. Let me clear this point in diagram. This is the diagram of bud stage. The purple substance attached with the basal membrane is new columnar cells, while in the middle they have polygonal cells. Now, as we know that the epithelium of dental lamina separates the ectomesenchyme by basement membrane having a certain swelling in on that membrane, about 10 points. Because we have 10 teeth in each arc. The surrounding mesenchymal cells proliferate the result in the condensations of two areas. The area of condensation immediately below the enamel organ is a dental papilla. Dental papilla is present just below the basement membrane. The ectomesenchymal condensation that surround the tooth bud and dental papilla is a tooth sac. This tooth sac is visible or well defined in the is not visible or not well defined in a bud stage, but become more defined or prominent during the cap and bell stage. The cells of dental papilla uh, form the dentin and pulp, while the dental sac forms cementum and periodontal ligaments. Now, the second stage of tooth formation is cap shape. As the after the bud shape, the tooth does not expand uniformly. It does not expand uniformly into a large sphere. Instead, unequal growth in different parts of the tooth bud lead to the cap stage, which is characterized by the shallow invagination on the deep surface of the bud. Now let me clear the concept of outer and inner enamel epithelium. The peripheral cells of the cap stage are cuboidal, cover the convexity of this cap, are called outer enamel epithelium. The cells in the concavity of the cap become tall columnar cell and represent the inner enamel epithelium. Now the outer enamel epithelium is separated from the dental sac and the inner enamel epithelium from the dental papilla by the delicate basement membrane. Means they are separated from each other by the basement membrane. It is the boundary line. Now stellate reticulum, polygonal cell located between the outer and inner enamel epithelia. As the result, the polygonal cells become star-shaped but maintain contact with each other by their cytoplasmic processes, which is a thread-like networks through which they are attached. As these star-shaped cells form a cellular network, they are called stellate reticulum. These networks are called stellate reticulums. This is the rough diagram of cap stage. After the cap stage, we move on to bell stage. As we know from the name that it resembles the shape of the bell, so we call it the bell shape. And this is the last stage of bell stage. Bell stage is further divided into early bell stage and advanced bell stage. Due to continued uneven growth of the enamel organ, it acquires a bell shape. In bell stage, crown shape is determined. It was thought that the shape of the crown is due to the pressure exerted by the growing dental papilla cells on the inner enamel epithelium. Now the pressure, however, was shown to be opposed equally by the pressure exerted by the fluid present in the stellate reticulum. The folding of enamel organ to cause different crown shapes is shown to be due to different rates of mitosis and difference in cell differentiation time. Now, inner enamel epithelium. The inner enamel epithelium consists of a single layer of cells that differentiate prior to amelogenesis into tall columnar cells called ameloblast. These elongated cells are attached to one another by adjunctional complexes laterally and to cell in the stratum intermedium by desmosomes. The cells of the inner enamel epithelium exert a strong influence on the underlying mesenchymal cells of the dental papilla, which later differentiate into odontoblast. Stellate reticulum. The stellate reticulum expands further due to continued accumulation of intracellular fluid. The star-shaped cells having a large processes and anastomose with those of adjacent cell. As the enamel formation starts, the stellate reticulum collapses to a narrow zone, thereby reducing the distance between the inner and outer enamel epithelium. Means the distance between the inner and outer epithelium has been reduced. After the inner enamel epithelium, let me explain the outer enamel epithelium. The cells of the outer enamel epithelium flatten to form low cuboidal cell. The outer enamel epithelium is thrown into folds which are rich in capillary network. This provide a source of nutrition for the enamel organ. Before the inner enamel epithelium begins to produce enamel, peripheral cells of the dental papilla differentiate into odontoblast. These cuboidal cells later assumes a columnar form and produce dentin. Advanced bell stage, characterized by the commencement of mineralization root formation. The boundary between the inner enamel epithelium and odontoblast outline the future dentino-enamel junction. Formation of dentines occur first as a layer along the future dento-enamel junction in a region of future cusps and proceed pulpally and apically. After the first layer of dentin is formed, the ameoblast lay down enamel over the dentin in the future incasal and caspal areas. Thank you.